Thanks for being here. It's actually going now. Jane.
Hey, can you all hear me okay? Great. I want to introduce Elinor. Elinor is actually a really, really good friend of mine as well, so I'm so pleased to be actually introducing her. Elinor's history is actually really interesting. She joined the Broad in 2002, which was actually the same year I joined the Broad as well. And she came in as a data analyst. She actually went back to school to get a Ph.D. To really kind of explore her passion on comparative and animal genomics, and actually came back to us, but without really leaving us, because you were always affiliated, right? To now head up the Vertebrate Genome Group and also the Karlsson lab here. So welcome, Elinor, and I'll be happy to take questions at the end.
Thank you very much. Hopefully, if there's any problems with the slides, I can now blame it on the fact that I made it in Keynote and now it's in PowerPoint, so just so you know. Thank you, everybody, for coming. So I'm going to talk about our dog project, but just a caveat at the beginning. I've never owned a dog. I don't know anything about dogs. When I started all of this, I got drafted onto the dog genome project as these things happen at the Broad, and all of a sudden I had to start figuring out what these pugs and border collies and all these things were. So I really come into this from the point of view of dogs being really interesting genetic models, and only after that really got to start to know them as kind of animals. So cats are great. Cat behavior, also very interesting. We're not doing that yet, but I really one day we're going to. But this is what we're actually studying, are these guys. So I'm not going to talk about our wolf project, but that is in fact a wolf on the left and a dog on the right. These puppies, especially the ones on the right, were these puppies that we swabbed every day when they were really, really little, so they thought that saliva swabs were just about the best thing ever. Apparently the biggest problem was trying to get them to only have one puppy chew on one swab. But to start this, I'm going to go way back into the mists of genomic time, back at the beginning in 2005, which is when we finished, actually here at the Broad, sequencing the dog genome. Back then, you actually got to write a whole paper and have the cover of a journal for sequencing one genome, which was pretty amazing. And we had a lot of fun with it. We actually got permission to Photoshop in Tasha, the dog that we sequenced, into the famous photo of Watson and Crick for the cover, so that was pretty cool right there. And yeah, so we sequenced a dog named Tasha. We did it here at the Broad, back on the old Sanger sequencers. Dog genome, very much like the human genome. It's a little bit smaller, about 2.4 billion bases, and it's got about the same number of genes. Dogs, humans, not that different. And I put this slide in here. I was going to take it out since I was talking to the genomics platform, but I figure you guys can correct me if I get any of the numbers wrong. But we're getting really, really good at sequencing genomes. The first human genome took like 13 years, and today at the Broad, it's about one genome every 12 minutes, which is a huge amount of data that we're producing.
Didn't that change to 7 or something crazy like that recently?
7 minutes?
Yeah, who came up with that number?
You guys have to be careful with this, because we're going to start expecting to get our genomes back 7 minutes after we give them to you, so which would be really cool. So anyway, we're getting really, really, really good at producing data to the point where, you know, on the Broad servers, we keep on trying to figure out how to get rid of data, and we don't even bother looking at the stuff from a few years ago because it's so small compared to the stuff we're generating now. But the problem we have, as most people in this room are going to know, is that even though we're getting really, really good at sequencing DNA, we still don't really understand what very much of it is actually doing. The human genome is about 3 billion bases long, and only about 1.5% of that are actually coding for exons in genes. We're pretty good at figuring out what the coding regions are, but you know, for example, 80% of the peaks that they're finding in these big genetic studies of human diseases are not in coding regions. And so we're trying to figure out how we actually interpret those peaks and understand what effect they're having on biology. And so comparative genomics is a way to do this. If you compare a whole bunch of genomes from a whole lot of species and you find a position in the genome that hasn't changed for a very, very long time, that's usually an indication it's doing something important. If you think about it, DNA randomly mutates. That's just what DNA does. And so the only reason you wouldn't see changes at a position in the genome is that if individuals that had mutations at that position didn't survive and have children. And so conservation is a way that we actually have of actually figuring out that probably somewhere around 10% of the genome, the actual sequence of the bases, is important to the function of the genome and the health of the organism. And as I said, most of that stuff is not going to be in genes. It's going to be regulatory stuff around it. And so pretty much all of the research that we're doing in our group is really focused around this question of how do we find the important parts of the genome, and then once we've found them, how do we actually figure out what they do? And to be honest, probably a very large part of the Broad itself is focused on those questions. This is one project, not the dog project. This is our 200 mammals project. I put this in here because we were having this meeting today, and I was talking to Jeremy, and he said he'd gotten the data back from our meerkats, but that somebody in the platform had thought it was really, really bad data because it aligned really badly to the reference genome. But they were using the reference genome for dog, which totally explains why it aligned poorly. But by the way, we work on tons of things other than dogs. I thought it was pretty funny, actually. It wasn't a major error, but I was like, oh, right. Yeah, we do send a lot of dog stuff through the platform, so we probably need to be really clear when we're actually using a different genome. And I think probably a good, so we've done 130 new genomes for the 200 mammals project at this point in time, and I think, what is it, about a third or half of those went through the Broad platform here, and then the other part went through Sweden. So that's worked really well. But what I'm going to talk about today is one of our favorite model organisms, dogs, and what we're doing with them. So dogs are really interesting because they're not wolves. They're actually very behaviorally, and in some other kinds of ways, distinct from wolves. And we think that dogs probably diverged from wolves somewhere between 15 and 40,000 years ago. That happened either in one domestication event or in more than one domestication event, and that it probably happened in Europe, Africa, the Middle East, Central Asia, or East Asia. So we're not really that sure about that yet. Anyway, dog breeds, which is what everybody gets really focused on dog breeds when we think about dogs, they're actually very young. So as far as we can tell, dog breeds, which is kind of when they basically said these dogs get to be in this breed and no other dogs get to be in this breed, so they closed the breeding population, are basically around 100 to 200 years old. And they're kind of defined by very strong selection on the way the dog looks and the color of the dog and things like that. There's very limited genetic diversity within the breeds. Basically within a breed, all the individuals are cousins of one another or worse. And there's very high rates of particular diseases in those populations. So as far as the genetics of dogs, we've actually focused a lot on these breed populations because they're interesting for all of these diseases. But recently, we've actually gotten much more interested in this piece in between. So there's actually 15,000 years or more there where we're basically breeding dogs to live with us. So they're adapting to our food sources. And then at some point in time, we decided to start breeding them to have useful behaviors. So we ended up with populations with different behaviors. And so this is pre-breed, but it's actually really interesting selection going on in this time. Part of the reason we were focused on the breeds was that we didn't need, because of the limited genetic diversity, we didn't need a lot of information in each dog and a lot of genetic information from each dog in order to be able to do gene mapping studies. As the slide at the beginning of this talk showed, getting genetic information from an individual is no longer really the challenge. And so that's part of the reason that we can start looking back in time a lot further. So today we have these populations that to various degrees have been enriched for certain behaviors. And at the same time, we have a lot of behavioral disorders in dogs that are shared with humans, like aggression, anxiety, cognitive dysfunction, compulsive disorders, phobias, impulsivity. And so the question is, and actually you see that a lot of breeds that are bred for particular behavior, like the herding dogs, will actually tend to have high levels of noise and storm phobia. So there seems to be some interaction between things we're breeding for and behavioral disorders that are occurring in these populations. So we have this project called Darwin's Dogs, where we're trying to find out how changes in DNA are actually affecting dogs' behavior and health. And the way I always describe it is basically, we had this study where we're looking at compulsive behaviors in dogs, but I was like, it would be really cool to be able to find out the place in the DNA, like how do you actually change DNA to get a dog to obsess over retrieving a ball? Like it's a normal behavior, but it's kind of remarkable that we've actually selected on changes in DNA that lead to that. This is an entirely gratuitous movie of cute puppies. These are some dogs that we're working with for our working dog project. So these are potential future guide dogs, but they're also very cute puppies. So how are we actually doing this with dogs? Actually, so there were, as you notice, 2 different colors of dog in here, so it kind of ties into the next slide. So we're basically going to take 2 populations of dogs, controls and cases, and look at where they're different. So we get a bunch of dogs, we do a saliva swab, and we test a whole bunch of markers across the genome. That moved. That was supposed to cross out the 900,000, not underline it. We used to be around 1 million SNPs max. Now we're using a sequencing-based approach as opposed to a genotyping-based approach, and we're at about 10 million markers now that we're getting per dog, which is pretty cool. And then we basically go across the genome and ask, is the SNP correlated with my trait? Are the cases and the controls different at this position in the genome? There will be many places where you see differences between two different dogs. What we're looking for here is basically a position that is consistently different between dogs that have a trait and dogs that don't have a trait. So the first one we studied, which is actually here at the Broad back when we were doing the dog genome project, was this coat color in boxers trait. The dog we sequenced, Tasha, was actually heterozygous for this trait. So she had one solid allele and one white allele. And because of this, back then, sequencing genomes was really, really hard. We wanted to find the mutation that was underlying this trait. So we knew that somewhere in Tasha's genome, by sequencing her, we captured both the normal version of this gene and the altered version of this gene because we knew she was heterozygous. As it turned out, that may not have been the best strategy because she was indeed heterozygous, which caused a major error in the assembly, and it took us forever to figure out what the hell was going on. But there was a good idea behind that to begin with. So we took the 9 solid boxers who have 2 copies of the normal allele and 10 white boxers who have 2 copies of the white allele. And interestingly, the white boxers, so this is one of those white phenotypes, is actually connected with deafness. So about 2% of the white dogs are deaf in both ears. And we did what I just said. We went across the genome. We looked at every markers across the whole genome and looked for a place where the cases and the controls were different from each other. And I think this was, I forgot to uncover the p-values for this, but it was actually about, I think it was 10 to the minus 11 or 12 actually that we hit with this top peak here. Clearly it's different. And it's over this gene called MIDF. And the reason I keep talking about this story, even though we published it back in 2008, is it's a really nice illustration of the regulatory changes that we're looking for when we look at diseases in both people and dogs. So it turns out MIDF is a very important gene, but it comes in different isoforms. So if you start making your protein at this dark blue exon here, which is called the A start site, use all the exons from then on, you make a version of the MIDF protein that's absolutely critical for healthy eyes. And we know that these white boxers, their eyes are totally fine. However, if you start making the protein down here at the M start site, you get a version that's very important in terms of normal pigmentation. And that's obviously what's been affected in these white dogs. Interestingly, they can make pigmentation just fine as adults because they can have spots, but they don't seem to have normal development of pigment when they're embryos. We knew we couldn't have any coding mutations in this gene because these exons down here are actually used in the A version as well. So if we'd had damaging mutations in the coding regions itself, we would have ended up with individuals with bad eyes. And so it turns out that when we narrowed down the region by adding in another breed, we actually did hit this region just upstream of the M promoter, the M start site, and we eventually managed to get backs. This is not going to mean anything to half the people in this room, but backs from each haplotype and sequence them and identify what the differences were in Tasha in that region, and eventually narrowed it down to 2 or 3 candidate mutations. And actually, what I really like about this trait is if you actually look at the heterozygous dogs, it makes sense. So when you're developing, your pigment cells actually start on your neural crest, so along your spine, basically, and then they migrate around. And so if you have normal pigment cell migration, it's going to migrate around your whole body, and you're going to be totally pigmented. If you have one normal version and one altered version, the way this beautiful puppy here does, it basically makes it halfway round. So you have a solid back, but you have a white front. If you have 2 white versions of the gene, basically you have very minimal pigment cell migration. So you're almost completely white. If you do have pigment, it tends to be a large patch on your head because that's where the pigment cells go first. And then finally, it turns out that you have to have pigment cells in your ears in order to be able to hear. They have a different function in the ears. And so in a very small percentage of white dogs, there's so little pigment cell migration, you won't even see it getting as far as the ears. And those dogs are actually much less likely to have patches on their heads as well. And it also kind of illustrates why we kind of keep on pushing this idea of working in a natural model system, so an animal that's kind of in the world and mostly healthy. So they'd studied MIDF before, but when they studied MIDF in mice, they tended to do things like delete the whole gene. And in that case, or at least take out an exon or 2. And in that case, they got white mice, but it had all of these other effects too. They were deaf, they were blind, and for the most part, they were also dead, which is obviously not something people would have tolerated in their dogs. And so instead, they selected on regulatory variants. So they have dogs that are white, very rarely deaf, their eyes are totally fine, and important for everybody that actually owns them, they're not dead. And so we kind of got good at this. We went on and studied a bunch of other traits, the Ridge and the Ridgeback breed, this ALS and the Corgis. And we got pretty good at that. We could map these single gene traits. And of course, being scientists, we then decided to go on to something that was much harder. And so this is what we call OCD in dogs. It's actually not called OCD in dogs. It's actually called canine compulsive disorder. We don't know if they're obsessing because they can't talk. But the vets that I've talked to swear they would not be surprised if these dogs did also obsess. But it turns out that as far as we can tell, it's actually very similar to human OCD, and it's thought to be a model for it. So OCD in humans is defined as doing an otherwise normal behavior so much that it disrupts your quality of life. Dogs don't wash their hands. That wouldn't be normal for a dog, but they do things like spin or chew on themselves. This is a Doberman on the right, and he's doing this weird thing called flank sucking. Wish I could get the movie to run. Let's see if it goes. There we go. That's the trait we actually mapped, was that flank sucking in the Dobermans. Similar age of onset in humans and dogs. The more severe form in humans is early adolescence, and in dogs it's 6 to 12 months of age. It's highly heritable in both species. If your parents have OCD, you're at much higher risk of developing it. And it also, in both species, they use the same drugs to treat compulsive disorders, these SSRIs like Prozac, and it works just as badly in humans and dogs. So about 50% of people have some kind of response, and about 50% of dogs have some kind of response. And this also points to why we're studying behavioral disorders in dogs from a human health perspective. As anybody that's kind of interacted with the Stenley Center here has probably heard, we have a problem in psychiatric disease research in that we have not actually developed any fundamentally new drugs in decades. We've reformulated things that are out there, but no new mechanisms of actually targeting any of these diseases. And at the same time, many, many people with psychiatric diseases are not getting relief from the available drugs, or there's so many side effects that they don't like to take them. And so there's a real need to actually understand the biology underlying behavioral disorders and psychiatric diseases, because rather than, a selective serotonin reuptake inhibitor, which sounds to me like it's basically saying, let's change serotonin signaling across your entire brain, we might actually be able to target much more specifically what the problem actually is in one of these diseases. So let's see, there we go. So we did exactly the same thing we did with the coat color. We did a genome-wide association study, looked for differences between about 92 Dobermans who had been diagnosed with compulsive disorder to 67 healthy Dobermans. You can see it's a lot noisier than our beautiful pigment GWAS that I showed you, the white coat color one. This is because as you would expect, it's a much more complex trait. There's probably many different genes influencing it. But we did get this nice peak on chromosome 7, which was over a gene called CDH2. So anybody who's worked in genome-wide association studies knows that it's always good to hit a gene because then it's much easier to actually tell a story. Most of the time we manage to hit regions without any genes that we can't say anything about. But in this case, we actually found a peak that sat right in the middle of this gene, CDH2. CDH2 is actually a neural cadherin. It's found in glutamatergic synapses as well as other types of synapses. And it's basically involved in learning and memory. So this is the protein that it encodes right here. And it basically stabilizes connections between presynaptic and postsynaptic synapses. And this actually tied in really nicely with early results that were coming out of a genome-wide association study in people. They're still working on the GWAS of OCD in humans because they're having trouble getting significance. But they found a bunch of genes that were also in these glutamate signaling pathways. But the other thing that came out of this was actually we also discovered a problem with studying dog breeds and doing studies within single dog breeds is that, as you might remember from the beginning of this talk, there's very limited genetic diversity within a dog breed. And that means you get regions of the genome where all of the dogs within a breed will look exactly the same. And so we actually took a look at those regions in the Doberman pinschers, and we found out that on chromosome 17, the second biggest region of fixation, as we called it, in the genome was right over this gene called CTNNA2. And that actually encodes the protein that's involved in anchoring CDH2 to the presynaptic and the postsynaptic membranes. And we went on and looked at 3 other dog breeds that had compulsive disorders and did see that variants in this gene actually differed between the cases and the controls. And it kind of points to the fact that the reason we were studying Dobermans was that they tended to have a very high occurrence of compulsive disorders in that breed. But one way that you might end up with a very high rate of compulsive disorders in a breed would be if every single individual in that breed carried a risk factor for it. And I think that's probably what's going on here. So we can't find CTNNA2 by looking in the Dobermans because the cases and the controls are going to look exactly the same. They're probably all carrying whatever the risk factor is. And the other problem we had was that we had about 180 dogs in our study, and that just isn't enough. We got that one peak. It was a beautiful peak. But that's only explaining somewhere between 5 and 10% of the risk of compulsive disorder in these dogs. And we needed a way to get up to actually having a big enough data set that we could find the other things that were going on. And as I said, it's very complicated. There's many, many different genes involved. This was a cool picture that Hyun-ji in our group made a few years later for a paper where she'd also looked at human genetic data from people with compulsive disorders and then looked at what had been found in mice and just found that it wasn't necessarily the same genes in each of the species, but they seemed to be all targeting the same pathways. So we marked the human genes with a little human head, the dog genes with a little dog head, and the mouse genes with a little mouse head. And we went into it thinking, we'll find the same genes, it'll be amazing. And then, after we didn't find the same genes, I decided to interpret this as being a positive, because if you think about it, if what you're trying to do is understand the pathways that are involved in something like a compulsive disorder, having a second species where you're finding a set of genes that is also involved in that pathway, but are different from the ones that you find in humans, might actually give you a more complete story about what's going on. Also, around the same time, this paper came out from many people at the Broad and around the world, which was the first GWAS, genome-wide association study of a human psychiatric disease that really, really reached statistical significance for a lot of different peaks in the genome. It's a beautiful study. Did a GWAS just like we had done for OCD in dogs. But in order to actually get that statistical power, they needed about 37,000 cases and 113,000 controls. So it wasn't that surprising that we hadn't actually gotten a lot of significance with 180 dogs. So we came up with this idea that even if behavioral genetics is much easier in dogs than it is in humans because of all of the artificial selection, and probably natural selection as well, that's been exerted on these traits in dogs, we're still going to need a lot of dogs to actually study something as complex as behavior. But at the same time, we were thinking back to that story I told you at the beginning about those 15,000 years between domestication and the modern breeds. We'd always really focused on studying these purebred dogs in order to understand things like behavior. But if the selection on behavior predates the modern breeds, it goes back, people have been using dogs for things like hunting and herding and farming for much longer than the dog breeds have been around. There's no reason that we should have to look at purebred dogs in order to study the genetics of behavior. And that's how we ended up with this project that we've now renamed to Darwin's Ark. We originally called it Darwin's Dogs, where we decided to just ask everybody to sign up their dogs. And it's amazing, we had these two different, the old strategy was we'd talk to people, they'd be like, I have a dog, I really want to participate, and we'd be like, what breed? And they'd say the wrong breed, and we'd say, we don't want your dog, which was just a huge bummer for everybody. Instead, we have now a project where somebody comes up to us and says, I have an amazing dog that I totally want to sign up for your study, and we just say, that's wonderful, come and sign up, we want them all. So it's actually been a lot of fun as well. So you can go to our website, if you have a dog, you should totally do this. It's darwinsark.org, and sign up your dog and join our project. It's built on this model that a lot of different projects at the Broad are really pushing, which is this kind of open data approach. I have this idea that if we can just generate a really large data set and let everybody have access to it, then people that are really, really good at manipulating data are just going to come and have some fun with it. And so we tell people that this is basically an open data project. So we don't own the data, we're not going to sell the data, and we will give access to this data to any researcher out there, and owners can also have access to the data. And if they want the VCF file for their dog, we're happy to give it to them. We'll probably give them the BAMs as well, although it's a 1X sequence to begin with, so I'm not sure what they do with it. Anyway, so you can go to your website, you can sign up. Mentioned at the beginning of the talk, I don't have dogs, but I really wanted some screenshots of the website. So I signed up my cat, Darwin. Also went and told Jessica in my group, who's been handling the database, to make sure we noted that as being a fake account and not a real one, so we didn't use it in our analysis. So I signed up Darwin. This is a new feature on our website that we're really excited about, this thing called Order DNA Kit. So we had this project where we said, come sign up your dog. We'll send you a kit and get a saliva sample. We did that. We've got like 20,000 plus dogs signed up. We've got 5,000 DNA samples. We have no money to sequence 5,000 DNA samples. People get really upset that we're not sequencing their dog. We're really upset that we can't sequence their dog, but unfortunately us being really upset doesn't make grant money magically appear, so this has been a bit of an issue. So we now have an option, if you want to, you can actually support your own dog sequencing, and we offer it basically at cost. We also have a super fancy version where we get a 30x genome, which is, we're actually going to be working with the Broad Genomics platform on it and constantly bugging them to make sure that our samples stay on time, because we're really curious to see how many people actually want a 30x sequence of their dogs, because it'd be a beautiful data set. And then we ask you lots of questions about your dog. So does your dog enjoy life? We always put the dog's name into the question. This is very helpful, both in terms of personalizing it and making sure that people that have more than one dog don't forget which dog they're answering the question for. And then we tell you how your dog compares to other dogs. Your dog enjoys life. Your dog seeks constant activity. Oh yes, this one is very popular. Some people love it. Some people hate it. I have no idea what it means. It'll be really interesting to see if any genetic correlations come out for that particular question. Darwin's name did not show up on there because I answered that he really, really didn't think that he was a dog. Which is a space down there to actually print him down there. And as I said, we set it up and we signed people up really, really fast. As of yesterday, we had over 21,000 dogs signed up, and people had answered nearly 2.3, there's about 200 and something questions per dog, and basically people tend to answer large numbers of them. So it's been going really well. We've got a big spectrum of different dogs. Oh, this is Nellie the Beagle. She's kind of a mascot for our lab. Hilarious dog. And we actually have, as I said, mutts, purebreds. We'll take any dog. So we look at morphological traits. This is partly because some of these traits have been mapped already in dogs, and we wanted some positive controls in there. We look at artificially selected behaviors, so retrieving, pointing, even something like your dog seeks companionship, which might be something that they bred for, say, in lap dogs and things like that. We're looking for the really serious, complex traits and disorders, things like we have some kind of published questionnaires that have been well validated around impulsivity, personality, things like that. And then we have this fantastic set of traits. I have no idea if anything's going to come out of this, but we basically sat down with a group of people that work as dog trainers and dog behavior professionals and said, we're looking for traits that are genetic or heritable, and we define that as you seem to see some difference in the prevalence by breed, that are easy for owners to identify and that people aren't going to care about. So they're not going to try and train their dog to do it or train their dog not to do it. So we're basically trying to maximize genetics and minimize environment. And so we have this fantastic set of questions around whether your dog does that, you know, the adorable head tilt, like they're confused, places the paw on other people's feet. This was their top scoring one. As a scientist, it never would have occurred to me to ask anybody whether their dog turned in circles before pooping, but apparently there's a chance that that's heritable. And then licks the empty bowl after finishing food. This one's actually interesting. We took a quick look at it, and it might overlap with some of the other eating behaviors, which could be quite interesting. Oh, and then we added on food allergy-related traits as part of the Food Allergy Science Initiative here at the Broad. So we've got some early genetic results from this. We've just started doing the analysis. We switched from doing genotyping to doing low-coverage sequencing kind of halfway through the project, so we've been getting everything up and running. But we now do have the kind of first GWAS that we're getting out of it. This is actually a new plot as of yesterday, so very excited. Kathleen, a graduate student in my group, kind of went out of her way to get this done. But we now have 983 dogs with surveys and genetic data, and about 9.5 million SNPs total in our GWAS. And we have one question about the size of the dog. We tried sending people measuring tapes. People don't know how to use measuring tapes. So we stopped sending people measuring tapes. Now we just ask them, how tall is your dog when they stand next to a person of average height? Ankle high, calf high, knee high, thigh high, or hip high, so 5 levels. And that's the GWAS we get out of it. It works beautifully. So the red line is the genome-wide significance level that's used in human GWAS, which is a p-value cutoff of 5 times 10 to the minus 8. And then these are all the peaks that are exceeding it. So we're seeing those blue ones, those are all genes that have been reported as associated with size before in dogs. The red ones are all things that we're finding for the first time in this study. We've got this L-coral gene, which has actually been attached to size in horses. PRKG2 is actually a dwarfism gene in cattle, so it does also seem to have something to do with size. Of course, the other interesting thing about this is it highlights some of the weaknesses of this approach. All we ask is, how tall is your dog? And so we don't really know whether it's a short, stubby, like dwarfism, corgi-type dog or a really, really small dog. or whether it has a short snout. And so we're actually getting genes for all those different things in here. And we're going to have to figure out whether we can ask additional questions to try and resolve what the phenotypes it's controlling. And that's just plots. Oh, it's just showing, this probably doesn't mean anything to anybody in here, but now that we have this really dense data, we have these really nice little peaks of each region, which I was just really cool to see for me. So that's just showing that these are all of our association scores, and that's the peak over the gene IGF-1, and the same thing down here. So this is all of the serious science that we're doing in the dog project, very important disease-related stuff. But most of the people that come and sign up for our project are not actually signing up for it because they really want to help psychiatric disease research. They're mostly coming and signing up for it because they want to know something about their dog. And often it's because they have a mixed breed dog and they want to find out what's in their dog. And so this project kind of started with what we called, of course, the Mendel's Mutts project, Darwin's dogs, Mendel's Mutts, which was basically we went and found 21 dogs who we had absolutely no idea what their backgrounds were and sequenced them and tried to figure out what breeds were in them. So for those of you that haven't really thought about pedigrees and things recently, this is basically what goes on with a mutt. So imagine you've got two purebred dogs. This is one of their chromosomes. They've got 2 copies of each chromosome, one from mom and one from dad. So the golden retriever has 2 blue golden retriever chromosomes. The border collie here has 2 red border collie chromosomes. And then they have a puppy. This is actually a golden retriever border collie cross because you can find anything on the internet. And so you can imagine you then take your first generation mix and you cross it with a poodle because people cross everything with poodles. And then you make more puppies. What happens? So you might imagine that you basically get this. So this puppy gets 1 copy of the chromosome from his parent, and he happens to get the golden retriever copy, and then 1 copy of this other chromosome from his other parent, so the poodle copy here. Not quite. So this misses that critical step called recombination. So on average, there's 1 recombination per chromosome per generation, and that actually just swaps around 2 pieces of the chromosome. And so your puppies are actually getting 1 chromosome. This one that he's getting from this parent here is actually mostly golden retriever with a little bit of border collie stuck on the end. And then you make another puppy, and the same thing happens with the break in a slightly different place. This is actually happening. There's also recombinations on the green poodle one. I'm just not showing them because they're both poodles, so you wouldn't see it. And so you basically do this generation after generation. And so what you actually have in mutts is this tiling pattern of different ancestry from different breeds going across each chromosome in large chunks. And so what we're trying to do is basically going across the genome of each mutt, and for each piece of the genome, say, which breed do you match the closest here? And so we have these 21 dogs. A bunch of them actually belong to people at the Broad. My sister's dogs are in there. It was basically if you ran into me within this 2-week period where I was looking for mutts, I'd planned to have 10. But then I was informed by the genomics platform that my sequencing now was half as much as I thought it was going to be. So rather than save all that money, I decided to just sequence twice as many dogs. Brilliant decision on my part, I think. Anyway, so this is the way the breed calling algorithm works. This is Nellie, that lab mascot I mentioned, a beagle. Nellie is in fact a beagle. We have a very confident 88% of her genome that we matched to beagle. And then we have a little bit that we can't figure out. And then a couple of other breeds show up, but they're both, bloodhound might actually be related to beagle in terms of its breed history. So it's possible that it actually is just a region that matches both, or matches bloodhound in Nellie slightly closer than the beagles we have in our data set. There's always a piece that's not matched. We're hoping that'll get smaller as our data sets get bigger and better, but it'll be interesting. It's basically either, it could be stuff that, in this case, it's probably just beagle that we can't make a confident match to beagle for. Algorithms like I'm not totally sure. But in the mutts, it could be ancestry from populations that we don't have. This is a golden doodle named Hubble, and Hubble is in fact roughly half golden retriever and half poodle. But this is what the mutts look like, and I was not expecting them to be this mixed. So this is Skylar. Skylar was adopted by Linda, who used to be in my group in California, and she was told that she was a lab pit bull mix. She does in fact have Labrador in her, down here. And there is some Staffordshire Bull Terrier, which could be confused with American Pit Bull Terriers. It might be that pit bull thing that they were talking about. But actually, her top breeds are Dalmatian Rottweiler and Golden Retriever, which are not things that anybody had expected to see. I also hadn't expected, you know, when you run into a mutt, everybody's like, what are the 2 breeds that are in this dog? You know, it's a this and this, or a this and that. And every single mutt nearly that we looked at had, you know, somewhere between like 6 and, 15 different breeds contributing really detectable ancestry. So these dogs are really mixed up. I stuck Frances in here. I really liked Frances because I met her owners at the Somerville Dog Festival after we had the results. And so I pulled up her results and I was like, I don't know what happened with your dog. I mean, for half of Frances, we did a pretty good job. Like we got Maltese, China Crested, Havanese, small white fluffy dog. I was like, totally makes sense. But like half of Frances, we don't have a clue about. And she's like, oh, Frances is an adopted street dog from Africa. I was like, well, that would explain it. So it turns out that Frances is probably half somebody's small white fluffy pet and half African street dog. So that was very reassuring to me. So this is Huey. So we have a Broad Ignite project where we're looking at wolf-dog mixes. And so I stuck Huey in here. Huey is basically a wolf-dog hybrid mix. So he's about 50% wolf. And I probably just totally messed up the experiment by telling Brittany, who's analyzing the behavior, what's going on with Huey. Ignore it. Okay, good. And then he's got German Shepherd, Alaskan Malamute. And actually, the people that own these wolf-dog hybrids are really fascinated to find out what the dog part of their animals are. What really interested me when I found out about the hybrids is that he's actually a 50-50, but a lot of them have very variable amounts of ancestry from wolf and dog. And then this is Lucky. Lucky belongs to Jeremy in my group. Jeremy gets very upset at us when we tell him that his dog is a Shih Tzu. But we're like, we can't argue with genetics. Shih Tzu is the top breed that's in Lucky. I mean, come on. Total Shih Tzu. But a whole bunch of other things going on. And we've been playing around a bit. We're not totally confident in this yet, but we're working on doing this chromosome painting stuff. So when we go across the genome, we're assigning ancestry at each region. And we think we can actually take that ancestry and also take a look at the variants that are connected to traits and figure out why dogs look the way they do. And so if you look at Lucky, he actually, at the dropped ear locus, which is right here, he has toy poodle ancestry, which makes sense. He actually does have dropped ears or hanging down ears. He has this EPAS1 is a high altitude adaptation coming in from the Tibetan Lhasa Apso. I'm almost completely convinced that we will never know whether this benefits Lucky at all. Because Massachusetts not being particularly high altitude. And then he's also got the furnishings, which is like the mustachy thing coming in from the Lhasa Apso. Also has this brachiocephaly mutation, but then I was like, his nose isn't that short, and then I looked it up, and it turns out that it's like one of many different risk factors. So we've been doing that for a while. The newest thing we have, and which is the reason why everybody in this room who has a dog should totally go and sign up, so we finally actually started reporting the breed results back to people, which has actually been a really long, challenging process because we had to figure out how to explain the genetic information to them in a way that was understandable and not upsetting. For example, if you had what you thought was a purebred Great Dane, and we gave you results back saying that your Great Dane was 93% Great Dane and 7% unknown, there would be a very large proportion of people that would get extremely upset at us for telling them that their Great Dane was not purebred Great Dane. And so we had to figure out a way to both kind of show the data as it is, but also explain it in a way that would not kind of create that illusion. And so this is what we came up with. It's a little hard to see. I need to make a version of this with bigger text. But this is a dog named Lucy. I believe this is Jessica's dog. Is that right?
No. You just asked me for a purebred, so I found purebred.
Okay.
And this is Goldens, because they're cute.
They are cute. We also do a lot of work with Goldens. Anyway, so we have right at the top of our breed results here, we actually state Lucy is a purebred golden retriever. So for anybody that's worried about that, that's what we're saying here. But we do show that there's actually a portion of the genome that we can't actually match confidently to golden retriever yet. We should get better over time. And then we came up with a few statistics we could show. So mean posterior probability is basically how confidently we can match Lucy to golden retriever. Obviously, we're really confident that most of her genome is matching really confidently to golden retriever. We spent about 20 minutes trying to come up with another word or term for mean posterior probability, because it sounds really complicated, and we couldn't come up with anything that we agreed on. So we were just like, it's science, we're just going to put it on there and people can deal with it. Genetic diversity is a euphemism. If you click on the little question mark, it'll tell you more, but that's basically the heterozygosity in the individual, so how different mom and dad is. Lucy's actually not too bad for a purebred dog. She's about 23% heterozygosity. The most diverse genomes that we're seeing are about 60% of the positions that are thought to be heterozygous that can be variable are variable. This was actually an interesting one because every once in a while we actually do see mutts that are super, very low genetic diversity within their genomes. So they have very little heterozygosity, suggesting that even though they're mutts, their parents probably weren't that unrelated from each other. So it is actually an interesting metric. And then finally, the percent of the top breeds right here. And so if you had to say, so Jenny is in fact Jessica's dog, got that one right. Jenny is a mutt. And so we basically say we conclude that she's a mixed breed dog, and then we give the different percentages of the breeds that are coming in. And we're actually pretty confident that we've got the breeds that we are calling, we're pretty confident in, but there's a bunch of her genome that we're just not calling very confidently at all. And finally, in the breed calling results, we have this critical piece that we're really excited about, where we ask people what they think of our results. So we just say, do you think we got this right? And if they say no, we ask, can you explain how we got it wrong? Scruffy's a purebred, but you got the breed wrong. He's not a purebred. If we said he was a purebred and he's not, they can choose Scruffy's not a purebred dog. He's a mix of breeds, but not these ones. Or he's pure, but we said that he was a mutt, and they actually think he's a purebred. Or it just doesn't seem right, in which they get a comment box and they can tell us why it doesn't seem right. This was partly because we wanted to make our breed calling algorithm better over time, so we wanted to figure out the ones that we were just not getting at all. But actually in the first week, we had a woman contact us to tell us that we were saying that her dog was a mutt when it was actually a purebred Cavalier King Charles Spaniel or something like that. Turns out there was a sample mix-up in the processing of the sample, and we'd actually gotten 2 samples swapped. So that was actually a really nice check on our system. We put in an extra kind of controls in place to make sure it doesn't happen again. But that owner feedback actually ended up being really helpful very early in the process of identifying that sample mix-up. And then finally, the kind of last thing that we're working on here is we're trying to link breed to behavior. I would have really liked to show you one of our nice GWAS plots, but those are just coming off of the coming off the computers now, and they'll probably change. So I didn't want to get into that yet. But Jessica, who's we already talked about, postdoc in the group, did this really nice analysis looking at our Labrador Retriever ancestry and our mutts a couple weeks ago, and showed that if you look at how much Labrador Retriever is in a mutt versus how people answered the question about whether their dog is super interested in retrieving things, you can see that as dogs become more and more interested in retrieving things, the percent Labrador Retriever ancestry in those dogs is going up as well. And so there does actually seem to be a connection here between the ancestry of the dogs and their behavior, at least in this one case. The other really fun thing about this is that we were then like, as you do in scientists, you're like, okay, what problems are people going to find with this? And the most obvious one is that people see in dogs the behavior that they're expecting to see. So if they know their dog is a Labrador Retriever, they'll be more likely to say he's super into retrieving things than if he's not a Labrador Retriever. But luckily, we'd actually just finished this other thing called the Mutt Mix Quiz, where we asked people for 30 dogs to come to our website and guess what mutts they thought were in these dogs. It's basically a question of, can people actually do this, say which breeds are in which dogs. The website's actually still there. You can go test yourself if you want to do it. It's called muttmix.org. But within this 2-month period, I think we got nearly 35,000 people to participate in this survey, which gave us 14,000 guesses per dog as to what their ancestry was. And what we found out is that people who consider themselves dog professionals are not any better than the general public, and basically everyone's terrible at this. So people are not perceiving behaviors in the mutts because they know what breeds are in them. And then finally, the last thing I'm going to talk about now is a project we were actually just getting started on, the genomics platform that's basically working with new technology that's being developed in the genomics platform, and with Victor Adelsteinson and his group, but basically expanding our focus in Darwin's arc from just behavior to actually looking at health and cancer as well. Dogs and humans get many of the same kinds of cancer, with the caveat that actually there are cancers in humans like angiosarcoma, which are extremely rare. They're actually very common in some dog breeds. So it's an opportunity to study rare cancers in humans on a scale that's not possible in humans. And so we basically have a new grant that we're working on where we're looking at cell-free DNA sequencing in dogs to actually detect the tumor's DNA in the blood samples from dogs. And then we actually want to look at response to treatment, and we actually want to get an exome capture off the ground. And then we're also going to be doing a direct-to-patient recruitment, kind of on the model of Count Me In here at the Broad, where you're actually sending kits out and getting samples from owners, with the idea that they're really engaged and invested in helping make this science happen. And we're also going to be getting a lot of other information from the owners as well. And this is a project that Kate McGuire, has been a grad student in my lab for a long time, but then got her Ph.D., so now she's a postdoc. And Con and Victor Adelsteinson and then Corey Painter is actually involved. It's a really cool kind of cross-institutional study. And so we did actually do some cell-free DNA sequencing with you guys. It worked beautifully. This was a dog with osteosarcoma. This is basically showing as you go across the genome, looking for deletions and duplications and copy number variants, basically. And you can see about 16% of that sample was tumor, and you can see all of the changes going across it. Here's a lymphoma. You can see the same thing. And then angiosarcomas, we're seeing the same thing. This top one was actually somebody at the Broad. We put out a call in the broadcast and asked people if anybody had a dog that had been recently diagnosed with cancer and wanted to get us a blood sample to try this out on. And then we're also doing a pilot study, because this is a question they've been really interested in answering in people that they haven't been able to yet. We're just basically looking at how cell-free DNA changes over time, over short periods of time, and also in response to treatment. And so just to wrap up, this was where I started with. But basically, the kind of focus of this is understanding what we can do with all of these different kinds of species that we actually share these really close relationships with. The reason the dog project works is not just because the dog's genetics are interesting, but because they live with humans, and humans know so much about them. They can take them for medical care, et cetera. And we actually rebranded from Darwin's dogs to Darwin's Ark for a reason. We're really hoping to actually expand outside of dogs at some point in the future to all of these other cool animals that we live with. And then finally, this got mixed up. By the way, I didn't talk about our tick project, but if you have ticks and you want to send them to us, you should go get a kit, project@akari.org. And then also, if you have your own dog, you should come and sign up. And I tried to mention the people as I went through my talk, but here's the full set of acknowledgments. So thank you.
Elinor, did I see you chickens?
Not yet, but we totally could.
For those of you who don't know, I am a crazy chicken lady. Happy to take questions for Elinor. Anybody? Did I see Hershey up there, by the way? Yeah, okay, I actually saw a GP dog up there.
Oh, really? Which one? If I'd been really organized, I should have messaged ahead of time and found out who the GP dogs were so I could have put more of them in. It would have been cool. Oh.
So this is kind of a selfish question because I'm the owner of Hershey and we got him sequenced through you guys. So how can we get that data? How can we help you more with other data that we might have? Is there anything that we can do?
You can get the data hopefully by sending us an email, and we'll get it to you. We're hoping to actually eventually have a system where there's basically a link and you can click on it and you can download your data. You should pester. Wait, is Jeremy here? No. You should pester him endlessly about it, because we've been talking to him about how to actually make that happen. So yes, it's that distinction between reality and ideal world. The data is all there. We're very happy to share it. We just need to get the infrastructure in place to make it easier, but sending us an email, we can get it to you that way. We should have the breed results on your page now. If they're not there, you should tell us, because that would be a problem. And actually, I think if you've answered all the surveys, we don't have anything else we need right now, but I think what we're going to be trying to do over the next few months is get this kind of health portion of the site up and running for the cancer project. And we'd be really interested to have people take those surveys and then also let us know when they're confusing, because asking people about whether their dog has a cancer is actually complicated, because you need to figure out which words you should use. You can start by asking them, has your dog had cancer? And then start asking them about what kind of cancer. But the different words that people use to describe diseases makes it a much more complicated survey to set up. And so we're actually going to hopefully, we can get some people to help us make sure we're doing that right. Also, get all of your friends to sign up and then pay for the sequencing. That would be so cool.
So I have a question. So we've been working closely with 23andMe doing some genome sequencing for them, and I was chatting with them, and they said that they have received dog samples as part of their studies. My question to you is, how many human samples have you received where people think they might actually be getting a cheap GWAS study?
As far as I know, none. Just so everybody knows in here, it wouldn't work. Wait, did you have a... We've received 2 fecal samples. Brittany is the one that has to actually open the packages when they come in. Yeah, and she's like, ooh, this is a really stinky one. I don't know how to open it. So yes, it's amazing what people will send you in the mail. Luckily, our kits are now going straight to the company doing the low-pass sequencing for the low-pass sequencing part of this, so they're going to have to deal with it.
Great talk. I have a question. So there's this famous experiment by, I think, some Russian guy who bred foxes to be more docile, and he succeeded in making them more docile, but at the same time, the ears became floppy. And you showed that the locus in dogs for floppy ears has been mapped. I wonder if there's any connection.
Yeah, you know, we've actually been poking into that a lot because it's a really intriguing hypothesis. And it's a little unclear exactly what's going on with the foxes because the phenotypes that they report being connected with the behavior are actually fairly rare in the populations. It's not like it's all of the foxes. At the same time, it seems totally plausible that, you know, if you had a locus that was controlling behavior that happened to be close to something that controlled appearance, that you might actually see changes in the 2 things at the same time. And actually, Jessica, the new postdoc in my group, is a new postdoc in my group because she just came off of a Ph.D. where she's actually working with brains from those foxes, actually looking at differences in the, is it the hypothalamus? Did I get that right? Pituitary. There we go. The pituitary between the more tame and the more aggressive foxes. But it's a really interesting study of just really strong selection on behavior in populations. You need the mic. I'm supposed to stop you until you have the mic.
So you had that slide looking at how people were bad at identifying dog breeds. Did you look to see if certain breeds were guessed correctly more often than others? Like they got it correct 25 overall, but maybe Labradors were really easy to identify as got right 50% of the time, and others
yeah. So we haven't dug into that in great detail yet. We do. So if you want to be right, and this is why we haven't actually finished the statistical analysis, if you want to be right, your best bet is to guess Labrador. Because if you actually look at the mutts, the percentage of each of the breeds that show up in the mutts is very well correlated with how common that breed is in the US dog population. So the more common the breed, the more likely there is going to be ancestry from that breed in the mutts. And this is why we had a 2-month study period for the Mendel's Mutts project when we didn't tell anybody things like that and then closed at the end so that we wouldn't bias our results. You can still do the surveys, it just won't actually go into the data set. But I'm actually, you know, at first it was just to answer that question of how often do they get it right. But now that we have, on average, 14,000 guesses per dog, we could start looking at that. The other thing I'd be really interested in looking at is whether people guess certain breeds based on particular physical characteristics. So if the dog has floppy ears, are they guessing some breeds more than others? Because I suspect there's a small number of physical cues that people are using to actually make these guesses about what breeds are in dogs.
I have a question. Thank you for your interesting talk. So will the consortium collect digital health information and the lifespan information of dogs in the future?
It's collecting which kind of information?
Digital health.
Oh, yeah, so we've actually talked a lot about that. So this is the idea that we could actually use the electronic medical records from veterinary clinics, like we use electronic medical records in humans. And yes, that's something that's definitely on our horizon and for the cancer project that we'd like to start working on. From what we've heard from people that have actually started working on this problem already, working with electronic medical records from veterinarians is very similar to doing it in human medicine. In both the good ways and the really bad ways. So it's a really complex problem where people use different codes for things and enter different information in different ways. But at the same time, I think that at least half, maybe the majority of veterinary clinics in this country are collecting information electronically at this point in time through a relatively small number of platforms. So it's something that could potentially work really well.
How about the lifespan parameters? Like Professor George Church are studying using dog for reversing aging research.
Yeah, no, I've talked a little bit with those guys. There's a lot of interest. So we're actually working with somebody who's launching a project, hopefully soon, looking at dog aging. And it's a really interesting model. It's the same reason why studying cell-free DNA in dogs is so exciting, because things in dogs happen faster. So we can do a 10-year long study in dogs and actually capture most of the dog's lifespan as compared to humans, where by the time we finish the study, we'd all be dead because it's human aging. But it's the same thing for cell-free DNA. If you want to look at something like, what do they call it, minimal residual disease, so seeing whether you can detect the recurrence of cancer once they've actually been treated. So actually detect relapse and how early you can detect it. That happens much faster in dogs on average than it does in humans, and actually in some of the breeds in a much more predictable way. So you could actually take a group of golden retrievers with lymphoma, and then once they'd had chemotherapy and gone into remission, just do a blood sample every month or every 2 weeks, because most of them are going to relapse within kind of a year time period. So it's a much more efficient way to do aging-related studies, or time-related studies, I should say.
Well, Elinor, thank you so much. I think we're going to wrap up, and thank you for presenting today.